Thank you, Catherine, and good afternoon, everybody. It's a great pleasure to be here and be able to provide an update on the Tadalafill Phase 3 study. I think this is the third PPMD meeting that we've attended, so I guess we're a relative newcomer compared to some of the other speakers, but I think it's just been incredible to see the amount of progress that's been made even in just the last 2 or 3 years. But again, I'll be talking about our ongoing Phase 3 trial of Tadalafill for muscular dystrophy. First, a few quick disclosures. Obviously, I am a full-time employee of Lilly. Tadalafill is currently only approved in adult indications, and I'll touch on those in the next slide. And this presentation is only meant to be for exchange of scientific information. So Tadalafill is a member of the class of drugs known as phosphodiesterase type 5 or PDE5 inhibitors. And I'll talk a little bit more about that on the next slide. Tadalafill is currently approved for 3 different indications. So under the brand name Cialis, it's approved for the treatment of erectile dysfunction, and also under the brand name Cialis, it's approved for the treatment of benign prostatic hyperplasia. Under the brand name Adcirca, it's approved for treatment of pulmonary arterial hypertension, which is basically a condition of high blood pressure in the arteries that supply the lungs. As I said, it's currently only an investigational drug for DMD, and it's not indicated in any pediatric population. Now, this is just a cartoon that kind of explains what the cellular mechanism of a PDE5 inhibitor is. And so this is just a cross-section of a blood vessel where you have the inner lining of the blood vessel being the endothelium, and then a large part of the wall of a blood vessel, especially an artery, is composed of vascular smooth muscle. So in the endothelium, you have an enzyme called nitric oxide synthase, which can synthesize this molecule nitric oxide. And what this molecule will do is diffuse to the smooth muscle, activate guanylate cyclase, which then produces cyclic GMP, and then through several different mechanisms, the cyclic GMP will cause a relaxation of the vascular smooth muscle and vasodilatation and an increase in blood flow. Now, like in most regulatory pathways and pharmacological pathways, you have an on switch, and the nitric oxide can sort of be thought of as the on switch in this pathway. But then you also have an off switch, and the off switch in this case is the PDE5. And so the phosphodiesterase 5 enzyme works to break down the cyclic GMP that's produced and therefore acts kind of as a limiter to this signaling pathway. So if you have a phosphodiesterase 5 inhibitor, that can amplify this cyclic GMP pathway, leading to vasodilatation and an increase in blood flow to affected tissues. And this is thought to be the mechanism through which these PDE5 inhibitors work for their approved indications. Now, how this might be involved in muscular dystrophy, we just need to talk a bit about blood flow regulation in muscle. And so in resting muscle, the blood vessels that supply blood to the muscle are normally held in kind of a constricted state, and it's done so through action of norepinephrine in the sympathetic nervous system. Now, when muscles undergo work through exercise, there is a muscle-specific nitric oxide synthase enzyme that gets activated, and this releases nitric oxide into the tissue, into the surrounding vasculature, which increases cyclic GMP, causes the blood vessel to dilate, and you get an increase in blood flow, oxygen supply, and nutrient supply to the working muscle. And it's thought that this feedback loop actually plays a very important role in the ability of working muscle to compensate for the increase in oxygen demand when it undergoes work, to compensate with that as for an increase in oxygen supply. Now, it's been known since the mid-90s that this nitric oxide synthase isoform in muscle is actually associated with this large complex of proteins that are associated at the sarcolemmal. And I know you saw some similar cartoons such as this at the session yesterday. And what will happen is with the lack of dystrophin that occurs in Duchenne muscular dystrophy, this entire protein complex dissociates from the cell membrane, and along with it, the nitric oxide synthase. And so you get a mislocalization of the nitric oxide synthase as well as a reduction in the expression of of the enzyme. So in that setting, this feedback loop that's normally present to increase the oxygen and metabolite supply to exercising muscle is dysfunctional. And so when that's dysfunctional, you can have the onset of functional muscle ischemia, which can then lead to cell damage and cell necrosis. And so this is thought to be at least one of the mechanisms through which, in the setting of muscular dystrophy, you have the progressive muscle dysfunction and muscle damage that occurs. And this is really the physiological basis that first suggested the idea or the hypothesis that a PDE5 inhibitor, by affecting this NO-cyclic GMP pathway and increasing cyclic GMP, may be able to sort of short-circuit this pathway and amplify the vasodilatation that needs to occur upon muscle exercise. Now, some of the first clinical experiments that were done to try to test this idea were done by Dr. Ron Victor and his group at Cedars-Sinai in California. And the first study that they did was actually done in adult men with Becker muscular dystrophy, and they found that single doses of Tadalafill were able to help alleviate the muscle ischemia when the muscles undergo exercise, and also increase blood flow in response to exercise. And those data were published in 2012. Dr. Victor then did a follow-up study in a small group of boys with Duchenne muscular dystrophy and basically found the same results, that single doses of Tadalafill were able to alleviate the functional muscle ischemia and increase blood flow in working muscle. However, whether these effects on muscle blood flow regulation and muscle ischemia have any clinical relevance with regard to slowing the progression of muscle disease in DMD is completely unknown, and it remains unknown today. And that's really the impetus and the rationale for doing this phase 3 trial that I'll talk about next. So just a background on the program. So we really came into the picture in late summer or early fall of 2012 when we were first contacted by a small group of DMD research and advocacy community members, including Pat, regarding our interest in collaborating on a phase 3 registration trial of Tadalafill. So after reviewing all the data that was available at the time, the decision was made to proceed with this. So for the remainder of 2012 and for the most of 2013, a lot of people did a lot of work to try to get all the prerequisite steps that are required before initiating a phase 3 trial done as quickly as possible. And so with that, we had the first patient enrolled into the trial in September of 2013. And then just this past January of 2015, we completed enrollment in the trial with 331 patients randomized. So this is a graphic of the overall study design as it was originally designed. I'll talk a little bit later about a recent amendment to the protocol that we did. It doesn't affect this part of the study, but I'll talk about that in a minute. You see the screening and randomization criteria. So obviously males with DMD, we don't have any specific mutation requirements for the trial. Ages 7 to 14, normal cardiac function by ejection fraction of at least 50%, ambulatory, and all on stable corticosteroids. If eligible, the boys were randomized to one of 3 treatment groups, either placebo or one of 2 doses of Tadalafill. These 2 daily doses were chosen based off of the single doses that in Dr. Victor's study were shown to increase muscle blood flow in response to exercise. And then after this initial 48-week placebo-controlled phase, the boys have the option of continuing into a 48-week open-label extension phase where all the patients will be treated with one or the other dose of Tadalafill. And so the primary objective of the trial was to test the hypothesis that once daily oral Tadalafill for 48 weeks compared with placebo lessens the decline in ambulatory ability in boys with DMD. So it is a global study, so we have patients enrolled at 63 sites in 15 different countries, including North America, Europe and West Asia and East Asia. We also have study locations in Puerto Rico and Argentina. These are the study endpoint measures. So the primary endpoint is the 6-minute walk. The secondary endpoints include the North Star Ambulatory Assessment, some of the time function tests that are typically used in the other Duchenne trials, and then the PODC functional health questionnaire. And then from an exploratory standpoint, we're looking at the performance of the upper limb and pulmonary function tests. Now, we also have 3 exploratory sub-studies that are being done at a subset of the study sites. So at many of the U.S. sites, they are collecting cardiac MRIs. And the main point of interest here is this MRI measure of circumferential wall strain. And I think in some of the other talks here at the meeting, and certainly I know at some of the posters that were up, you may have seen some of the information around some of the emerging cardiac MRI markers that appear to be more sensitive perhaps than other more conventional echo measures of cardiac function. And in fact, they may start to decline at a younger age than something like left ventricular ejection fraction. So that's a particular point of interest in the cardiac MRI study. Also, as many others, we're collaborating with the imaging DMD sites who are also involved in the Tadalafill study, collecting lower limb MRI and magnetic resonance spectroscopy data to see if we can look at progression of the skeletal muscle pathophysiology by T2 and fat infiltration. And then at a single study, or a single site, at Dr. Victor's site, he's also doing some of these skeletal muscle blood flow measurements identical to those that he did in the early clinical experiments that served as the rationale for this trial to see if chronic daily dosing of Tadalafill will have a similar effect. Now, I mentioned the amended study design, and just to talk about what prompted this. So, as I said, we completed enrollment just this past January, and when we started to look at the timeline for how the enrollment occurred, we realized that some of the earliest patients that were enrolled into the trial would actually be completing the open label extension, the initial 48 weeks of the open label extension, for some months prior to when we would even have data from the placebo-controlled period available and have at least the initial answer as to whether or not the drug was working. So we decided to extend the open label extension for another 48 weeks. So now we have 2 periods of the open label extension where basically the amended protocol will provide for continued access of Tadalafill at least through the end of the double-blind treatment period and availability of the primary efficacy analysis. Under the scenario where the trial is successful, and if we do meet the primary endpoint, then continued access would continue through at least week 144, or 3 years to the date to where the patients were originally randomized. And the amendment would also provide, again, under the setting that the primary placebo-controlled phase of the trial shows efficacy, for collecting longer-term safety follow-up as well as information on ambulation and upper extremity function through a total of 3 years of follow-up. Just to touch on the current study status, so as I said, we started enrollment in September of 2013 and completed with 331 boys randomized in January. Looking at how the enrollment progressed, we're anticipating that the double-blind placebo-controlled portion of the trial will complete, we'll have the last patient visit in December of this year, and then hopefully have at least some initial results expected during the first half of 2016 and have, I think, an answer to this important question of whether Tadalafill may provide some benefit in slowing the progression of decline in walking ability. So with that, I'd just like to say some special thanks to obviously all the boys and their families for their continued participation and time that they have given to the trial, all the site investigators and staff that made timely completion of enrollment possible, Duchenne Connect and Treat NMD both helped us with enrollment and also contributed to completion of enrollment. And also a special thanks to the physiotherapist and physical therapist training team for all their expertise and dedication. This group spends an incredible amount of time traveling to the sites and spending time at the sites, making sure that the functional evaluations are being done correctly and according to the protocol, so we can be assured we're collecting the highest quality data as possible. And so I just really want to thank them and thank you for your attention.